Okay, welcome to the video for 9.1. We are going to be mainly talking about something that we call gene transfer. So how do bacteria share these genes that are going to help them survive when an environment changes? So let's talk about this. So we just got done talking about natural selection. We just got done talking about how some traits are going to allow an organism to fit into their environment better, which is going to allow them to survive, and it's going to allow them to reproduce and pass their genes on to the next offspring. Now, if we're talking in bacteria and we're talking about a selective pressure like a antibiotic, well, our bacteria are going to hope for a mutation that's going to create antibiotic resistance. Now, the thing is, if we're waiting for a random change in our DNA that's going to change a gene and automatically confer antibiotic resistance to our bacteria, this is not going to happen very quickly. This is going to be very slow. In fact, it's probably going to be a collection and an accumulation of random mutations over a longer period of time. So if we're just waiting for that mutation to happen, unfortunately, that bacteria is not going to survive that environmental change. It's not going to survive that selective pressure. So it's going to more likely than not go extinct. Now, if the mutation has happened and they already have the antibiotic resistance, they're not going to really feel that selective pressure. They're going to be able to survive and pass their genes on. But the thing is there a way that we can be faster at getting these adaptations? Okay, getting genes that are going to confer things like antibiotic resistance. Is there a faster way to get these? And the answer is yes, and this is what we're going to talk about in 9.1. So 9.1 is all about gene transfer, recombination, and mobile genetic elements. So the learning objectives for this section, you need to be able to explain how genetic information moves between bacteria by transformation, transduction, and something called conjugation. And then we're going to jump in and talk about gene mobility, so how genes move around the genome as it is. And we're going to talk about something called DNA recombination and how this can lead to the emergence of new pathogens. So the first thing we need to talk about is the different types of gene transfer. Okay, we've already seen vertical gene transfer kind of show up. This is literally what happens with us, right? Our parents pass the genes from them onto us. This is vertical gene transfer from parent to offspring. Now, this chapter is not about horizontal gene transfer, right? There's no parents there's no sexual reproduction in bacteria. Okay, so what we're going to look at is something called horizontal gene transfer. Okay, this is going to be the transfer of genes from one organism to another organism. Now, this is going to be non-offspring. Okay, this could be organisms that are the same species as the other organism, or they can be even different species. Okay, let me reiterate, this is not sexual reproduction. Okay, this would be like taking the gene from your dog for its ears and giving it to your cat. Okay, or vice versa. Okay, this is just taking single genes or groups of genes and transfer them, not the whole genome. Okay, we have four modes of horizontal gene transfer. Okay, these move whole genes between these different organisms. Okay, so this is going to result in rapid evolution. So we're going to see these bacteria species evolve very quickly. Okay, our four modes of horizontal gene transfer, and you need to know these for the next exam, and you need to be able to explain them. The first is transformation, which is where we're going to get DNA from the environment and take it up and integrate it into the genome if we're a bacteria. We have conjugation, which is going to be a one-way transfer of plasmids from one bacteria to another. We have transduction, which is going to be what we call virus mediated. So we have viruses that can infect bacteria, and they can actually move genes between different bacterial species. And we'll talk about this. And then finally, we have the good old jumping genes. We have what's called transposition. This is going to be those mobility elements. So we're going to see genes move from different parts of the genome. Okay, they can do this by either copying and pasting themselves or cut and pasting themselves out. Okay, so these are our four modes. Let's talk about them in a little bit more detail. So let's talk about transformation. Okay, so transformation is the process of bacteria importing free DNA into their cells and integrating them into their genome. Now, transformation can happen one of two ways. It can be natural or it can be forced. Okay, so a lot of bacteria can naturally find DNA fragments and plasmids in their environment from nearby dead cells and pick them up. Okay, so this is actually why it's important that we have started to not only take care of bacteria using chemicals, but we've started to use techniques that are going to degrade DNA in the hospitals, so bacteria that are in the hospitals can't pick up things that are going to be advantageous for them, like antibiotic resistance. Okay, so they can do it naturally just by finding a piece of DNA in the environment and picking it up and importing it into their cells. Okay, or we can force the bacteria to do this. Now, we do this in the lab all the time because we have learned how to harness some of the processes that bacteria have in order to benefit us medically. Okay, so there are some bacteria that do not import DNA very efficiently, but we can force it. Okay, there's two ways that we can force it. One is by electroporation. Literally, we shock the bacteria with electricity and convince them to pull the DNA into their cells. Now, this one can take a little bit of time. There's an easier way, in my opinion. The other scientists might differ in their opinions on this. We can use chemicals and heat shock in order to get them to do this. So one of the ways that we can do this is using a ionic salt like calcium chloride, which is going to make the membrane more porous. It's going to disrupt that plasma membrane. And then what we can do is we can heat shock the bacteria and get them to uptake the DNA this way. So when we heat shock bacteria, what we're going to do is we're going to take the broth and we're going to put it into an ice water bath. Okay, and we're going to let it sit there and get really cold. And then what we're going to do is we're going to remove it from that ice bath and we're going to put it into a boiling water bath, so a hot water bath, and raise the temperature up and then put it back into the cold water. This is what we call heat shock. Okay, so we're going to have the plasmids in the broth and we're going to take it and let it sit there for a few minutes, and then we're going to take that broth and we're going to incubate it at 37 degrees Celsius and see if the bacteria pick the DNA up or not. But this is how we do that. Okay, so a lot of times when we're talking about transformation, bacteria are going to naturally take up DNA from their environment. We use this all the time in biotechnology and medicine. In fact, I'm actually very, very, very grateful for this whole process because this is how we make human insulin now, is we use transformation to do that. So we use plasmids in order to get E. coli to make insulin for us. Let's talk about how we discovered that bacteria can do this. So back in the early 1900s, like 1930s, 1940s, we were trying to figure out what our genetic material was. What molecule in our bodies did it? Was it proteins? Was it lipids? Was it nucleic acids? Okay, was it carbohydrates? So we were going through a bunch of different experiments trying to figure out what our genetic material was. So there was an experiment that was part of this process, and it was run by Frederick Griffith in 1928. So Griffith was working with a infectious version of Streptococcus pneumoniae. Okay, so he's actually working with two strains and studying the disease process. So there's two strains of Streptococcus pneumoniae. There's the R strain. Okay, this is because the colonies, when you grow them, look rough on the outside. And then we have the S strain, which has smooth colonies, so they're not rough on the outside when we look at them. Okay, so the R strain and the S strain differ from each other. The R strain has no polysaccharide capsule on the outside of it, and it's non-pathogenic. Whereas with the S strain, it does have a polysaccharide capsule, and it's pathogenic, meaning it causes disease. Now, what Griffin was doing is he was injecting both strains into mice and seeing what was happening. So when he would inject the mice with the R strain, as we can see here in this graphic, the R strain is non-pathogenic, it's not virulent, it's not going to cause disease. Okay, so what's going to happen is it's going to leave the mouse living. Okay, it's not really going to affect the mouse. Now the S strain was very virulent. It's very pathogenic. It's going to actually kill the mouse. It's so virulent. Okay, so when he would inject the Sleuth strain into a mouse, well, the mouse was going to die. So what he did is he took the pathogenic version and he killed it and then injected it into the mouse. Now, once he killed it, it was no longer virulent. It was no longer able to pass along the genes that would make it cause the infection. Okay, so when he injected the Sleuth strain that he killed into the mouse lived. Now, not quite sure how or why he did this next part, but one of the things that he did is he combined both the living R strain, so the non-pathogenic version, with the heat-killed S strain, so the one that was pathogenic that no longer works. Now, when he did this, he saw something very surprising. Okay, so when he did this, he expected to see the mouse living, right? Because with the R strain, it was living, and with the heat-killed S strain, it was living. Well, when he did this, the mouse died. What's happening here? So what Griffith came to realize is that even though he had heat killed this Smook strain, this pathogenic strain, the R strain was still living. So the R strain had picked something up from the heat killed S strain that converted the R strain to the pathogenic version, so the version that can cause disease. Okay, so this is how we discovered transformation. Now, Griffith didn't realize that it was DNA that the bacteria was picking up, but he did realize that genetic material could be picked up and transferred between cells in this way. Okay, so we took something that was safe and we made it deadly. We gave these mice pneumonia. So we take this process of transformation today, and in the lab we use plasmids to take a gene of interest and study it, not just use it for biotechnology. Let's talk about how this works when it comes to human insulin. So diabetics cannot produce insulin on their own. There is a mutation that's going to affect the protein and make it so that it's not going to work right. Okay, so they can't control their blood sugar. Okay, so previously we used to take pigs and cows that were on their way to the slaughterhouses, and we would isolate the protein from these pigs and cows. Now, this was a very expensive process at the time, like hundreds of dollars to get this treatment. And we had some problems with it. So not all isolations worked. Sometimes we didn't get insulin from those pigs and cows because we did something wrong. And sometimes when we did, a patient would take the insulin and they'd have an allergic reaction, right? Because this is a completely different species insulin. So even though it was similar and would work, we'd get these allergic reactions. The way that we do it today is we're going to use E. coli in order to grow human insulin. So we have figured out the sequence of the insulin gene in the human genome. Okay, so we can make that in the lab, so we can have those bases together. And what we can do is we can take one of our plasmids from bacteria and use our endonucleases, our restriction enzymes, to open this plasmid up, and then we can stick the insulin gene inside the plasmid. Okay, so this is what we call a recombinant plasmid. Remember, when we take genes from one thing and put it into something else, we get recombinant DNA. That's not DNA that's normally found there. Okay, so our next step is once we have built this plasmid, we need to be able to get it into the bacteria so it can uptake that DNA, that gene, into its genome in order to produce the insulin. So what we're going to do is this is where we're going to force the bacteria to uptake the plasmids. So we're going to use that calcium chloride and that heat shock method to get the plasmid into the bacteria, and then the bacteria are going to integrate it into their genome, hopefully. Okay, there are some that won't do it, and they're not going to be effective in this process. Okay, so once we have that done, we're going to take our transgenic bacteria and we're going to grow it in culture. So we actually grow these in large anaerobic vats. Okay, so big tanks that we just grow all these bacteria in. So we're going to let it grow for a few replication cycles. We're going to get a lot of it made. And what's going to happen is we're going to take the E. coli from those vats and we're going to lyse the cells and isolate the human insulin protein from that mixture of lysed cells. Okay, and then so finally, we will get a nice vial of purified insulin that diabetics can then use for treatment. Okay, so this process was very expensive. This one, it's about $34 a vial in order to make insulin, which is very cheap. Now, what insulin costs from the pharmacy versus how much it costs to make are two different things. In fact, a lot of diabetics have a hard time paying for insulin because the companies are charging a very high rate for it. Okay, so we use this process of transformation to study lots of things in science. We have used it to make components of the COVID-19 viruses. We talk about in 8.6 how we can make a mouse glow. It's using the same process using that GFP protein, but we can also make bacteria glow. So GFP is not our only glowing protein, it's just the one that glows green. We actually have a bunch of different glowing protein genes that we've used to make different bacteria that glow different colors. In fact, there's this whole art movement in science. It's called microbial art, where we take these glowing protein genes and put them in the bacteria, and we use them on plates and make art from them. Okay, so we have green, we have red, we have blue, we have some yellow. Oh, excuse me, we have some pink. Well, last year's microbial art, there's always going to be a microbial art contest through the American Society of Microbiologists. This is the professional category. So last year's competition was space themed. As you can see here, we have these little alien guys. They're all different colors. Those are pretty cool. This is the one that won first place. I'm not the biggest fan of it. I liked this guy. Look at the detail that they got by simply growing bacteria that glows in the dark on agar. Also, this guy right here is pretty cool too. The whole Perseverance. So we have the Ingenuity helicopter, and we have Perseverance here. I think that's pretty cool. Now they have different categories. They have a kids category that's pretty cool. They have a creatives category along with the professionals. So I would just Google microbial art if you want to see more examples of this. People have done some really amazing things in this art style. Okay, so let's talk a little bit more about transformation, because we've been talking about in our examples about moving one gene for a protein into a plasmid. Well, plasmids can carry fully functioning multiple resistance genes. Okay, it doesn't just have to be one. Okay, so we can confer antibiotic resistance with a single plasmid to multiple antibiotics at once. Now, this is a problem in healthcare. This is scary. Okay, so if a bacteria species, so especially pathogenic bacteria that are susceptible to antibiotics, pick this plasmid up, they are now going to have these resistant genes that is going to make it so that the antibiotics that would kill it before no longer work. Okay, this is the whole basis of infection in microbes right now. In fact, we talked about the escape pathogens. This is the way scientists think they got their resistance. They were able to pick up these plasmids, these genes that conferred upon them these fully functional resistant genes. Okay, so the next type of horizontal gene transfer we're going to talk about is conjugation. Sometimes people refer to it as bacterial sex. It is not bacterial sex. There's no sexual reproduction in bacteria, right? It's all binary fission. So conjugation is the type of transformation that is going to involve direct cell-to-cell contact. Okay, the cells have to be in contact with each other in order to transfer genes in this method. Okay, so there's a special pili called a conjugation pilus. Sometimes it's referred to as a sex pilus. We're just going to call it a conjugation pilus. So the donor cells that are going to give plasmids to the recipient cell are going to attach to the recipient cell using the conjugation pilus. And then this protein tube, this pilus, is going to be how they send DNA to the recipient cell. So let's talk about how this happens. So let's talk about how conjugation happens. So we have our donor cell here. We can see its chromosome and we can see the plasmid. So this plasmid has what is called the F factor on this. This is going to be very important. So this is our recipient cell. We can see that it does not have the plasmid. So what's going to happen is the donor cell is going to extend its conjugation pilus and it's going to attach to the recipient cell. Now once it's attached, it's going to retract the conjugation pilus and it's going to bring the recipient closer to it, and it's going to form a protein bridge here. Now this protein bridge is going to allow the transfer of the plasmid from the donor to the recipient. Okay, so what's going to happen is an enzyme is going to come in and it's going to nick the plasmid genome, or the plasmid gene here, at ORET, and what's going to happen is it's going to allow the transfer of the plasmid through this protein bridge. Okay, so this is going to happen. It's going to be donated as a single strand, and once it gets into the recipient bacteria, its molecular machinery is going to come in and it's going to make it double stranded again. And as it's transferring, the donor's molecular machinery is replacing that single strand with a new one. Okay, so at the end, the recipient is going to now have the plasmid and have that F factor gene. Now, this is how a plasmid could be transferred between bacteria that will confer antibiotic resistance from one strain to another. Kind of scary and kind of cool. There should be a animation video on Canvas that you can watch about this as well that will go into a little bit more detail. But that's pretty much what we need to know for this class. Okay, so we've talked about transformation, we've talked about conjugation. Now we need to talk about the third one, which is transduction. So this is going to be virus-mediated genetic transfer. Okay, so with bacteria, we have viruses that are specific to bacteria. So they only infect bacteria. We call them bacteriophages. Okay, and just like all viruses, they are going to be a genome encapsulated in a protein jacket. It's called a capsid. Okay, with bacteriophages, they have this really cool design. They look like a lunar lander. Okay, so they're going to have what is called the sheath. This is going to act like a injector on a syringe. Okay, so they're going to attach to the bacteria cell. They're going to plant this injector into the membrane of the bacteria, and it's going to shoot the genome from the capsid through the sheath into the bacteria. And this is going to allow it to transfer genetic material to the bacteria. Okay, so this is going to also be a way that we can transfer genes from one bacteria to another. Now we have two types of transduction. Generalized transduction and specialized transduction. So in generalized transduction, what's going to happen is the bacteriophage is going to attach to the bacteria cell. It's going to inject its DNA. Okay, that DNA is going to be transcribed and translated. So we're going to start to make new proteins. We're going to start to make the different pieces of the virus. And we're going to have the host genome in there too, remember, because everything's just one large open space in bacteria. So what's going to happen is enzymes are going to be made that are either going to degrade and break down the host genome, or it's going to literally cut it on its own. So what happens is when the virus goes to be assembled to make new viruses, and what's going to happen is it's going to pick up pieces of the bacterial genome and load them into the capsid instead of the viral genome. So now we have all these new phages. Okay, so some are going to have the viral genome and some are going to have pieces of this bacteria genome. Okay, so what's going to happen is once the virus particles are made, the cell is going to lyse and these phages are going to be released and go to infect other bacteria. So if we get one of these phages that has the genome from the host bacteria, and it goes to inject that there. So instead of injecting the viral DNA, we can now incorporate this new DNA from that previous bacteria into this bacteria's genome. So this is another way that we can have horizontal gene transfer. Okay, so this is done by accident. This is not meant to happen, but this is one way that we can transfer genes by this virus. This is why it's called virus mediated. It's because the transfer is being done by the virus itself. So let's talk about the other type of transduction, this specialized transduction. Okay, so this differs in the fact that in the previous one, the phage DNA itself stays separate from the host DNA. So in specialized transduction, we're going to have our bacteriophage attach itself to the donor cell. Okay, it's going to inject its genome, but now instead of being directly used for transcription and translation, this genome is actually going to go and insert itself into the host genome. Okay, where it's going to sit until they need to make new viruses. So what's going to happen is this phage DNA is going to need to be removed from the host genome. And when it does this, it's going to take some of the adjacent genes with it. So we have this capital D gene, and what's going to happen is when we cut it out and take the viral DNA and put it back into the phage, when we make new viruses, we're going to bring some of the bacterial genome with it. Okay, so what's going to happen is we're going to have these new virions that have this mix of both virus and bacterial DNA. It's going to come to a new recipient cell. It's going to inject that into it. So see, our host cell has this little d gene. So it's going to inject this mix of viral and bacterial DNA. It's going to go and integrate itself into the bacterial genome. So now we have both the capital D and little d genes. So this is another way that we can transfer genes in between two bacterial cells. So the difference between generalized and specialized is generalized is just going to accidentally pick up some bacterial DNA when it degrades it, whereas with specialized transduction, it's literally going to integrate into the genome, and when it excises itself out of the genome, it's going to take some genes with it. Okay, and now this recipient cell is now going to be able to pass both of these genes onto its progeny. So when it goes through division, that gene's going to stay in there now. Okay, so how can a bacteria cell protect itself from bacteriophages, from having these viral genomes integrated into its DNA, or how does it just protect itself? So we're going to talk about those endonucleases again from chapter 8. Okay, we call these restriction enzymes. Okay, so restriction enzymes are a way that bacteria came up with to combat these viral infections from bacteriophages. So what's going to happen is the restriction enzyme is going to cut apart the viral DNA so it cannot affect the host's DNA. But how do we keep restriction enzymes from cutting the bacterial chromosome instead of the viral genome? What's going to happen is there's going to be other enzymes that are going to protect the places where the restriction enzyme can cut. Okay, so remember it uses palindromes. So this is what we have. We have GAATTC this way and GAATTC done this way. So it's the endonuclease is going to cut both these places and cut this into pieces. So what happens is there's another enzyme that's going to add what we call methyl groups, a CH3 group, to the bacteria. So the enzyme is called methylase. So it's going to come in and add these restriction endonuclease sites. It's going to add this methyl group to the DNA. And because that methyl group's there, the restriction enzyme can't come and bind and cut the DNA in those spots. Okay, so this is going to be what protects the DNA of the bacteria from these restriction enzymes. And now the only things that are going to be cut are the phage DNA that have been injected into that bacteria. So we have one more type of horizontal gene transfer, and we call these mobile genetic elements. Okay, they're what are known as transposable elements or transposons. Okay, these are genes that literally can jump from one place in the genome to another, or from the genome to a plasmid residing in the same cell. Okay, we have transposons in all walks of life. In fact, we think that transposons that we have in humans came from things like viral DNA that no longer is effective in our genome. So the virus integrated its genome into ours and left the transposons. Okay, so the process of transposons, these genes jumping from one place to place, is called transposition. Now, transposition can happen one of two ways, where we have a copy and paste into a new place in the genome, or we can cut a whole gene out and move it to a new place. So here in this example, this is one of the copy and paste examples. Okay, so we have our transposon gene here, and we have this site on the opposite side of the genome where it can actually bind. So what's going to happen is this enzyme called transposase is going to come in. It's going to bind to our transposon, our mobile genetic element, and then it's going to bend the DNA until it attaches to the target site, and it's going to copy this gene into this target site, so this other gene, and it's going to disrupt this gene, so maybe it doesn't work as well or only partially works now. But this is how it copies and pastes itself. Now, we're getting into the holiday season, especially around Thanksgiving, so you have all probably seen examples of transposition happen in some of our food. So an example of transposition happening in real life is with Indian corn. So corn actually used to be this dark pigmented color. So what happens is there are transposons in this Indian corn, in this maize, that is going to take the gene, it's going to take the transposon and it's going to insert itself into the gene for this pigment. So what we're going to see is these yellow kernels instead of these red ones. In fact, a lot of the corn that we grow around here are the ones that have this pigment gene disrupted, which is why we get this yellow color. And then you can also see these like speckled guys. This is an example of when the gene copied and pasted itself, but we still kind of have a functional gene for that pigment. So we see a little bit, but it's going to be differ between kernel and kernel. So this is an example of transposons in real life. Now, this is actually how we discovered transposons, is in Indian corn. What's really sad is that the scientists that discovered transposons in Indian corn, Barbara McClintock, had a real hard time in the scientific community because they just didn't believe her that genes could jump from place to place. In fact, it took years in order for the scientific community to actually recognize the fact that she had discovered transposons. And it was awful the way that they treated her. So let's talk about the other method that transposons can use, this cut and paste method instead of copy and paste. So this is a way that transposons can move between genomes and plasmids. Okay, so we're going to have this gene for tetracycline resistance. It's going to be part of a plasmid. Okay, this gene is a transposon, so it means it can jump between the plasmid and the chromosome, and that's not what we see. So it's going to cut the gene from the plasmid, and it's going to insert it into the genome for the bacteria. The plasmid is no longer going to contain that gene, and the gene's now in the bacterial chromosome. We have now successfully moved a gene for antibiotic resistance thanks to this transposon. So this bacteria and all of its progeny are now going to be resistant to tetracycline. Okay, that is 9.1. We've talked about the different mechanisms for horizontal gene transfer. I would definitely know each of these four and be able to explain them for the exam. So we will see you in the last section for chapter 9 and 9.5.